Well, hello. Our biology ventures continue in chapter 5. And the first part, the first section of chapter 5 is on the cell membrane, components and structure. And once again, I'm following the basic pattern of Clark, Choi, and Douglas, main authors, not the only ones, of Biology 2E out of Rice University, OpenStax. So it's free materials. And therefore, I'm assuming they don't mind for pedagogy sake that I have used their diagrams from their book. In some previous videos, I've also used some Wikipedia images. And of course, they've used some Wikimedia images. Well, let's move on. So the cell membrane. This is not easy stuff. In fact, I feel like it could be taught, surely, more easily than it's presented. But I'm going to do these videos, and we'll do our best as an amateur. So the cell membrane has two basic functions. On the one hand, it kind of defines the cell's borders, what's in the cell, what's not in the cell, and it keeps the cell functional. The cell membrane helps, for example, when it gets cold. Variations in the cell membrane can help the cell survive better, regardless of the temperature range and so forth. The fluid mosaic model is the model that has pretty much evolved over time. I'm amazed at how science has been able to identify kind of the nature of such incredibly small things. You can see cells under a microscope, but it took a while for scientists to be able to identify the cell membrane, because the cell membrane is what, about 1,000th the size of a cell itself. So we can see the cell under a microscope, but the cell membrane is 1,000th of a cell, and so I'm amazed at how the science has been able to do this. So the fluid mosaic model basically says that the cell is fluid. It's squishy. It's squishy. You're squishy. So the cell membrane is fluid, liquidy, semi-permeable, meaning it lets some things in but doesn't let other things in. But it's got a mess of stuff stuck together, and that's kind of a mosaic. Is like, for example, the kind of mosaics I think of little tiles of different colors and shapes and so forth. Well, another way that I might, as a complete non-specialist, refer to the cell membrane fluid mosaic model as the basically lasagna model. So there's your diagram from the book of the cell membrane, and we've got. Doesn't that look like lasagna? I'm not sure I want to have lasagna now for supper. I wasn't going to have it anyway. Who are we kidding? But you can see you've got these. We're going to go into more detail about this diagram. There are the microfilaments from a previous video that are kind of giving girth, from the inside. But you've got this kind of layers of, it's bilayer. Okay, so there's two layers here. This called phospholipid bilayer. So you got some with the balls on the top and the legs coming down, and then you've got the balls on the bottom and the legs going up. And these two layers of phospholipids, lipid fat, phosphate, so you've got that. And then you got stuff stuck in it. It's a spicy meatball, You've got your proteins. These are basically proteins of various kinds doing things. There's a twisty protein. And then you've got your these are cholesterol in the middle. It's all very fun, isn't it? And then these are kind of mixtures, a glycoprotein, carbohydrates, basically, sugars. So this is a sugar attached to a protein, and this is a sugar attached to a fat, a lipid. And these are kind of little trees, on the top. Okay, well, let's go into more detailed. So the phospholipid lasagna, this mess in the middle here. Okay, so you've got two layers, balls up and balls down, and they're facing each other. The balls are the more phosphate part. Molecules with balls facing down inside, So the balls face down, inside they're at the bottom, they face outside at the top. So this is actually in a sphere. And then, okay, so we're going to talk about hydrophilic parts of these molecules and hydrophobic. So if somebody has a phobia, I'm afraid of the dark, so the hydrophobic side of the molecule doesn't, I don't like water, I'm afraid of water. And then the hydrophilic, phileo in Greek means to love. And so the hydrophilic side of it, I love water. Come be with me, water. So the balls basically are the hydrophilic part of these molecules, and then the legs are the hydrophobic part of it. And it basically has to do with whether or not. So these phospholipids, the hydrophilic part, that's the ball that likes water, it's polar. Polar means it's attracted to ions. And so, but the hydrophobic in the middle is non-polar. So there are two sides to these phospholipid molecules. The one side, the ball side, is hydrophilic and is polar, and then the inside is hydrophobic and is non-polar. Okay, so here's a breakdown of these phospholipids. So there's the ball. The balls have a phosphate, PO, remember PO, PO4 with a -1 charge. So you've got phosphate at the top. Or these are three different ways of diagramming it. This is the simplest way of diagramming it. It's a ball with two legs, Dun, 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 dun. Well, you can tell by the way I move my hammer. Anyway, I didn't quite pull that one off. Okay, so this is the same thing, but with the balls of the molecules, and then this is a diagram part. So the phosphate with a glycerol, that's basically a glucose with 3 of the hydrogens replaced by OHs. Anyway, and then these are the fatty part, the lipid. So this is the phospho, and this is the lipid, and this is a saturated. So if it's maybe I'm throwing too much out here. Hold that thought. We'll come back to the saturated and unsaturated legs of the phospholipid. That looks funny. I just picture Dance Fever or walking down some 70s video. Okay, with flared pants, Okay, fatty acids. So these are the phospholipid legs. The legs. These are the lipid, phospholipid. The legs are the lipids. Lipid begins with an L. Leg begins with an L. And so these fatty legs, all the fat goes, all the fat goes to its legs. Thunder thighs. Sorry, hope no one is offended. Anyway, okay, so if they're straight, that means it's just straight hydrogens. No carbons with double bonds. And so this actually, from what I can tell from the reading, the saturated legs don't help as much in cold temperature. So the unsaturated leg, because it's angled a little bit, it's not only more attractive and seductive, look at that leg there, but also when cold makes it contract, these keep the cell from rupturing. It gives, again, more sturdiness to it. So phospholipid legs, the lipid part, saturated. If they're saturated, they're straight. And if they're unsaturated, that means there are some carbon double bonds in there and fewer hydrogens. Okay. Okay. So proteins. So we've got the base, the kind of the fundamental part of the lasagna are the phospholipids, either facing down or facing up. So the phospholipids, that's the heart of the cell membrane. However, there are all these blue proteins that are scattered throughout of different kinds. And remember, these are being produced by the ribosomes and sent through the endoplasmic reticulum to the Golgi complex and on up through Good grief. Ah, my brain is fried. Wizard of Oz, you know how the Glinda the Witch comes in a bubble, so the protein is taken up to the cell membrane and it lodges in there, different kinds of proteins. So what are these kinds of proteins? There are three basic kinds of proteins. You kind of see them. You've got some that are on the surface, inside or outside the surface. You've got some that are kind of giving girth, and then you've got these kind of tunnels that go through, and then you've got, I call these twisties. So you've got, first of all, you've got integral proteins. Integral proteins are integral. They solidify. You know how sometimes in a house you'll have poles or load-bearing walls that are kind of giving girth to the structure. That's my interpretation. These integral proteins, they go through the whole thing. They face on both sides, and there's a twisty one right there. So some of them are like plugs, and then some of them are like twisties, and some of them are like channels, protein channels that apparently let some stuff through. Those are integral. They're integral to the structure of the cell membrane. Then you have these peripheral membrane, the ones that are just on the top, and these kind of shake hands with other cells from the body. So the kind of like the bouncer proteins, they're standing at the door, show me your ID. And so these peripheral membrane proteins kind of alert, does not belong. So these things on the top, including the glycoproteins and the glycolipids, they kind of are security guards, on the surface to say, does this thing belong or does this not belong? And one of the problems with viruses is viruses can fool these security guards with fake IDs. Okay, so we've got glyco lipids, grief, my brain, phospholipids. We've got phospholipids, proteins, and then we have carbohydrates. So the carbohydrates are these sugars, glycoproteins, glycolipids, that again are kind of security guards. What is a carbohydrate? Well, it's in the formula of one carbon for every oxygen and then two hydrogens for each one. So this is an empirical formula because it multiplies. That's what the N means. The N means, so C2H4O2 or C3H6O3. So it can multiply out. Glucose is C6H12O6, for example. It's a carbohydrate. Glucose is sugar, right? So these carbohydrates can attach to proteins and to lipids and kind of stand like trees on guard there at the surface of the cell membrane. So glycoproteins and glycolipids. We call that kind of surface, not me, but biologists call that kind of surface of the cell the glycocalyx, which is basically all of that stuff, all the sugary, the lovely sugary mixture on the outside of the cell. Lastly, there's cholesterol hiding in here. Cholesterol is a fat, it is a lipid, also a steroid, which I didn't do videos on cholesterols yet. Tell me if you want me to go back and do some of those. But these lipids help in, for example, varying temperatures. They help provide stability to the cell membrane when it gets really hot or really cold. They're in the middle. They're within the tail bilayer. And that's one of the questions from a previous chapter, I think. I noticed I haven't been doing the practice questions at the ends of the chapters. I'm going to try to do some of those. But it is the cholesterols are in the middle, within the tail bilayer. So these are the tails, the lipid tails of the phospholipids. And that's a bilayer, and the cholesterols are in the middle. Cholesterols help with the fluidity of the membrane. They help keep it fluid, keep it flexible, because cells have to be able to move, right? They need to be able to kind of do a little bit of bending. And they also, as I said, cholesterols help with the temperature changes. Okay, so here are some questions from the end of the chapter that I probably shouldn't give the answers, but let me give you some of my thought process of how I would go about thinking about these questions. So, which plasma membrane component can be found either on its surface or embedded in the membrane structure? Well, let's do process of elimination. I just mentioned that cholesterols are in the middle, so it wouldn't be cholesterol because it's in the middle. Carbohydrates. Well, I know there are carbohydrates on the surface because that's where you have glycoproteins and glycolipids, but I don't remember the lasagna having any of those in the middle. I just don't remember that. Phospholipids. Now, the phospholipids uh, they span the whole thing. They do. But in that sense, they're not really on the surface or embedded. Because they're the whole thing. So again, you need to think for yourself. What do you think? But I think I would pick protein. And please feel free to disagree because I am not a biologist. But there are proteins definitely on the surface, and there are proteins that are embedded through it. And so that's what I would probably pick. But think for yourself because I can be wrong and often am. Why am I making these videos? Okay, so which characteristic of a phospholipid contributes to the fluidity of the membrane? Okay, well, I'm guessing, that's a good question. So a saturated fatty acid tail is straight. The double bonds, this would be unsaturated. That helps with temperature and with contraction. Well, what do you think? What do you think? Let's go back. Oh, cholesterol helps with the fluidity of the membrane. I'm going to go with B, cholesterol. What do you think? Don't just take my word for it. You have a brain. Okay. What is the primary function of carbohydrates that are attached to the exterior of cell membranes? So these are the glycolipids and the glycoproteins. Well, I'm thinking it's A, but double check, because remember, they're the security guards that are up there and kind of helping to identification, please. So I'm thinking that it's A. I don't know that it's strengthening. It's not a channel through the membrane. I don't think it adds to the flexibility. But do not take my word for it. You need to think for yourself. In fact, maybe I shouldn't do these questions. Okay, this one was hard. I really am not sure about this one. A scientist compares the plasma membrane composition of an animal from the Mediterranean coast with one from the Mojave Desert. Which hypothesis is most likely to be correct? The cells from the Mediterranean coast animal will have more fluid plasma membranes. Well, I don't know. The cells from the Mojave Desert will have a higher cholesterol concentration in the plasma membranes. I actually don't know if this is true because remember, cholesterol both are hot, right? Both the Mediterranean coast and the Mojave Desert are hot. The Mojave Desert is in the western United States, Arizona, New Mexico. It's dry and it's hot, but both of these are hot. And cholesterol helps with the fluidity of the membrane. But there's where the catch may be, because cholesterol does help with the fluidity of the membrane, and you might think that the Mojave Desert, being a dry place, would have more difficulty with the fluidity. So that could be it. C, the cell's plasma membranes will be indistinguishable. Yeah, I really doubt that, because dry and wet, I doubt that they'll be indistinguishable. And that's not a typical answer that a book would put on a test. D, the cells from the Mediterranean coast animal will have a higher glycoprotein content. while the cells from the Mojave Desert will have a higher lipoprotein content. If those are the identification ones, then I'm not sure about D. Well, I am leaning towards something now, having gone through these, but I'm not going to tell you what it is. Although you can probably guess if you've been listening, but you need to figure it out for yourself. I actually spent probably half an hour digging around trying to find an answer, but hopefully the fluidity is the same. You would think that's a goal, that it's going to adjust things within the cell so the fluidity ends up similar. Well, I'm going to leave it at that. I don't know the answer. I'm leaning a certain way, but I'm not going to tell you what it is. This has been yet another biology video by Ken Shank.